Hello everyone, my name is Janvi and we will study the subject biology in which we discuss the topic genetics and Mendlism. What is Mendlism? Experiments performed by Mendel on genetics and description of mechanism of hereditary process and formulation of principle are known as Mendlism. What is genetics? Genetics is collective study of heredity and variation. Genetics term was given by James Watson. What is heredity? It is a transmission of character from one generation to another generation, or we can say that parents to their offspring is called as heredity. Heredity is also known as inheritance. What is variation? During the transmission of character from one generation to another generation, there is change. This change is also called as variation. Let's see the picture of Gregor Mendel. This is the photo of Gregor Mendel. Fast facts or overview of Gregor Mendel. Gregor Mendel is known for scientist, friar, and abbot of St. Thomas Abbey who gained posthumous recognition as the founder of the modern science of genetics. Gregor Mendel is also known as John Mendel. He was born on 20 July in the year 1822 and he died on 6 January in the year 1884. He gained his education in the University of Olemus and University of Vienna. Gregor Mendel is also called as father of genetics. Let's discuss the brief life story of Mendel. First, Mendel's education and the Abbey of St. Thomas. John Mendel, he was not called Gregor until later, was born on July 20 in the year 1822 in the village Hazendorf bio Oradu. This small village was in the Australian Empire, but it is now the Swiss Republic. Mendel's parents were small farmers who made financial sacrifice to pay for his education. This is the birth house of Gregor Mendel, where he born in the village Hessendorf Biodrau. Mendel well enough at high school to make it to the University of Olemus in the year 1840. The university was about 40 miles from his home village. The distance from his home village to the university is 60 kilometers. When Mendel is 18 years, he took courses in physics, mathematics and philosophy. For better understand, we see the picture of University of Olemus. In the photo, we see university and garden. His teacher says to Mendel that you want to keep doing science, you need to be a monk. By the time he was 21, Mendel had run out of money. One of his teachers, the physicist Professor Friedrich Franz, advised Mendel to join the Abbey of St. Thomas in Brunn as a monk. By doing so, he could continue studying science and not starve. So Mendel, who was more interested in science than religion, became a monk. This is the photo of Abbey of St. Thomas. The Abbey actually had a good reputation for its teaching of science, and its director, Abbot Franz Cyril Knapp, was particularly interested in the heredity of traits in plants and animals on farms. The move to Brunn took Mendel about 80 miles from his home village. On joining the Abbey, he took the name Gregor. From then, he ceased to be John Mendel and became a Gregor Mendel. Learning and teaching science. In the year 1846, he aged is 24-year-old. Mendel took fruit growing classes given by Professor Franz Deibel at the Brunn in Philosophical Institute. Deibel was an authority on plant breeding. 1 year later, he went to the University of Vienna, where he studied chemistry, biology, and physics. The idea was that by strengthening his knowledge in these subjects, he could qualify as a high school teacher. After completing his study in the year 1854, he returned to the monastery and became a physics teacher at a school in Brunn, where he taught for the next 16 years. This is the photo of University of Vienna. This is second University of Mendel. Research and admin. In the year 1856, Mendel aged is 34-year-old. Mendel again failed to qualify formally as a high school teacher, this time because illness prevented him completely the exam. In the year 1865, still interested in physical science, he founded the Austrian Meteorological Society. In fact, during his life, Mendel published more papers about meteorology than he did biology. In the year 1856, he began his major groundbreaking study of heredity in plants. We see in the photo. In the year 1866, he published his heredity work, but unfortunately, most people who read it did not recognize the intellectual gold his paper contained. In the year 1867, his age is 45-year-old and he became abbot of his monastery and devoted himself to its smooth running as its administrator. At the monastery in Brunn in the early 1860s, Mendel is pictured back right, looking as a part of a plant in his left hand. Abbot Franz Cyril Knapp sits in the front row wearing a large cross. Abbot Knapp encouraged Mendel's science and heredity studies. Let's discuss Mendel and genetics experiment with peas in the year 1856 to the year 1863. During his time in Olemus, Mendel had made friends with two university professors, Friedrich Franz, a physicist, and John Carl Nestler, an agriculture biologist who was interested in heredity. Nestler passed his interest in heredity to Mendel, who was intrigued by the subject. Mendel's monastery had a 2-hectare garden, and his two former professors encouraged Mendel to pursue his interest in heredity by using the garden for experiments. Abbot Franz and Professor Franz Deibel also encouraged him to follow this path. This is the photo of Mendel's garden. Reason for Mendel's unsuccess, or we can say that limitation of Mendel. Mendel published his work on inheritance of characters in the year 1865, but for several reasons it remained unrecognized till 1900. Firstly, communication was not easy as it is now in those days, and his work could not be widely publicized. Secondly, his concept of gene or factors in Mendel's words are not acceptable. Thirdly, Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of the time. Finally, though Mendel's work suggested that factor genes were discrete units, he could not provide any physical proof for the existence of factors or say what they were made of. Reason for Mendel's success. Mendel studied the inheritance of one or two characters at a time, unlike his procedures who had considered many characters at a time. Selection material of pea plant. Selection of garden pea plant is suitable for the studies which have the following advantages. Pea plant is annual plant with short life cycle of 2-3 months, so large number of offspring can be analyzed within a short period of time. Second one, it has many contrasting traits. Third one, natural self-pollination is present in pea plant. We see in the left side of picture.
Cross pollination can be performed in it artificially, so hybridization can be made possible. We see in previous slide. Pea plant is easy to cultivate. Pea seeds are large. In addition to pea, Mendel's worked on rajma and havak weed. We see the picture of these seeds in further slides. Mendel's quantitatively analyzed the inheritance of qualitative characters. Fourth, he maintained the statistical records of all the experiments. Let's see the picture of different types of plants and their seeds. First, pea plant and their seed. In the left side, we see the picture of pea plant, and in the right side, we see the photo of pea plant seed. In one side, Rajma plant is located, and another side, Rajma seed is present. This is the photo of hawkweed. First one is orange hawkweed and second one is head of white hawkweed. Technique used by Mandel. He developed a technique emasculation and bagging for hybridization in plants. Flowers of pea plant are bisexual. In this method, one considers as male and another as a female. Removal of anther from a bisexual flower in immature stage is called emasculation. Emasculation is done to prevent self-pollination. Emasculated flowers are covered by bags. This is called as bagging. Bagging is only used to prevent undesirable cross-pollination. Mature pollen grains are collected from male plants and spread over emasculated flower. Seeds are formed in the female pollination. The plants that are obtained from these seeds are called first filial generation or F1 according to Mendel. The plant of F1 generation are self-pollinated and F2 generation is produced. Mendel took a pea plant and listed seven main contrasting characters. Seed in form Seed is in the form round and wrinkled. Cartilage in the form of cotyledon, yellow color and green color. Flower in form of color, white and violet. Pod in form of size, full size and contrasting sized. Color green and yellow. Stem plus axial pods and terminal pods. Size tall and short. Mendel result for flower color. Mendel found the same result for all traits, but we will look at flower color as an example. When Mendel bred purple flower peas BB with white flower peas small BB, every plant in the next generation had only purple flower, capital B and small b. When these purple flower plants, small b, capital B, were bred with one another to create second generation of plants, some white flower plants appeared again BB, small bb. Mendel realized that his purple flowered plants still held instruction for making white flowers somewhere inside them. He also found that the number of purple to white was predictable. 75% of second generation of plants had purple flowers while 25% had white flowers. He called the purple trait dominant and the white trait recessive. Punnett square. Each of staring plants have purple flowers, but they contain the genes for purple flower, capital B, and white flower, small b. The pollen grain from male plant fertilizes the egg in the female flower. In this variety of plant, purple flower are caused by dominant gene capital B. Dominance is indicated by capital letter. White flower are caused by recessive gene indicated by small letter. First time Puneet square was used by Reginald C. Puneet in the year 1875 to the year 1967. This representation of generation to another analysis in form of symbols and symbols of squares. Male gametes lie horizontally and female gametes lie vertically. Both the both the male and female parents plants in the diagram above carry the dominant gene B for purple and recessive gene small b for white flower. The ratio of purple flower to white flower in their offspring will be 3:1 as shown in this diagram. For a white flower to appear, the offspring must inherit the recessive gene from both parents. Purple appears with any other combination of gene inherited from the parent's plants. Mendel's law of inheritance and experiments. During his experiments, Mendel found that certain unknown factors are involved in the transfer from a parent to offspring. Those unknown factors are known as genes. Two main experiments were conducted by Mendel to determine the law of inheritance. First, monohybrid cross experiment, second, dihybrid cross experiment. First, monohybrid cross experiment. A monohybrid cross is a cross between two homozygous parents with different single pairs of genetic characters. In these experiments, two pea plants, one short and one tall plant, were crossed between them, where he received a tall pea plant in the first generation of offspring which he named F1 progeny. Then again he crossed the F1 progeny and received both tall and short pea plants in a 3:1 ratio. He even conducted his experiments between other contrasting characters like green pea and yellow peas, round peas and wrinkled peas, purple flower with white flowered peas, and many more. The results were found to be similar in all the cases, from which the law of segregation and the law of dominance were formulated. A phenotype is observable physical appearance of an individual that results from the interaction with a genotype. The genotype is a set of heritable genes carried by an organism that is transferred from parents to offspring. Homozygous is two copies of the same allele. Example: capital Y, small y, and heterozygous is the different form of a particular gene. Example: capital Y and small y. What is law of segregation? Mendel's law of segregation states that during meiosis, the two parents' genes separate and get divided into each gametes receiving per alleles. The hereditary factor segregates during gamete production so that offspring receive one gene from each parent. This is Mendel's third law of inheritance, while law of independent assortment is the second law and the law of dominance is the first. Law of dominance. Mendel's law of dominance is the first law which states that in a heterozygote one allele will dominate the other for the same character rather than contributing to a phenotype. The suppressed alleles are called the recessive traits, while the determined traits alleles are called dominant traits. These two recessive and dominant trait alleles refer to the genotypic interaction of alleles. Other researchers have found that the law of dominance does not rely on every time. Instead, several different patterns have found to exist. Dihybrid cross experiment. In a dihybrid cross experiment, two completely different organisms that are heterozygous are identically hybrid for two traits. It carries two different alleles. Mendel conducted his experiment on a pea plant that was wrinkled green seed and round yellow seed. After crossing these two homozygous and dominant for wrinkled R and green capital Y seed similarly, homozygous and recessive for the round small r and yellow small y seeds. He obtained the first generation progeny, F1 progeny, to be a mixed round yellow heterozygous plant, R, capital Y, and small y. Mendel again self pollinated the F1 generation through which he obtained a 9 is to 3 is to 3 is to 1 ratio with four different traits. The 9 is to 3 is to 3 is to 1 ratio described the 9 plants with round yellow seed, 3 plants with round green seeds, 3 plants with wrinkled yellow seeds, and 1 plant with wrinkled green seeds. Mendel observed in this experiment that the parental traits sorted independently from one generation to another, which was when he formulated his second law of independent measurement. Law of independent measurement. Mendel law of independent measurement states that a pair of traits separates independently of one another during gamete formation. Different traits of individual hereditary factors get equal opportunity to occur together. The biological selection for one trait does not depend on the selection of any other traits. Mendel found support for his laws by obtaining a 3:1 ratio result from the monohybrid cross and 9:3:3:1 ratio result from a dihybrid cross experiment. This is the photo of Punnett square and gametogenesis in F1 generation and in F2 generation. The ratio is 9:3:3:1. The result of his experiments were published in the school journal Nature for Scandinavian in the year 1865. A paper of Mendel by the name Experiment in Plant Hybridization was published in the journal. Rediscovery of Mendlism. At the time of Mendel's discoveries were negligible. It was rediscovered after many years. After 16 years of Mendel's death in 1900, Mendel's postulates were rediscovered. Rediscovery by three scientists independently. Karl Korensen is the scientist of Germany and he experiment on maize. Hugo de Vries, he is the scientist from Holland and he experiment on evening primaries. This is the photo of Karl Korensen and Hugo de Vries. This is the photo of E-Reach 1 test mark CSN egg. This is the scientist from Austria and he experiment on different flowering plants. At the end, Gregor Mendel was unaware of the new science of genetics he founded and unaware of any future controversies. And he died in the age of 61 of kidney disease on January 6 in the year 1884. Thank you so much for listening.